My name is Anna Korea. I work as an outreach coordinator and bioinformatic analyst on the BV-BRC project. I'm joined by my colleague, Dr. Christian Zamasic, and we are excited to show you our outbreak pages and then some phylogenetic analysis within the BV-BRC. So this is as I mentioned, we'll cover the outbreak pages and phylogenetics, and then we have a example with monkeypox virus to really showcase how you can utilize the BV-BRC for analysis. And there'll be some conclusions, and feel free to ask any questions along the way in the chat or afterwards, but feel free to stop us at any time with questions. So this is the second of 3 webinars. So next Thursday, we will have our final outbreak webinar for the fall, and that will focus on just BV-BRC analysis tools as a whole. So check that out. And before this, we had one on searching for BV-BRC data, and all of those will be loaded on our YouTube to watch whenever you feel like it. So if this is your first time interacting with the BV-BRC, we're happy to have you, and we want to highlight our mission. So we are here to provide a one-stop shop for data about human pathogenic and related bacteria and viruses into a scalable resource to help support comparative bioinformatics, large-scale data mining, multi-scale systems biology, and machine learning that will support the evolving needs of the infectious disease community and serve as a paradigm for research data resources. So the BV-BRC supports diverse bacterial and viral communities. We have a unified data model and real-time data integration process. So the format of data on the BV-BRC is very structured and similar. So each page will follow the same schematic so that you can feel comfortable no matter what bacteria or virus you're working on, you can understand where everything is. And we can support exponential growth of pathogen data. So we didn't used to have millions of SARS-CoV-2 genomes, but now we have tens of millions. So we can really support all of the data as viruses evolve and new outbreaks happen. So BV-BRC also pulls in consistent and accurate genome annotations, and we automate and manually curate the metadata to make it as useful as it can be for you. And then all of that data can be put into different gene or protein groups so that you can then run high throughput data analysis services on it. And we also integrate multi-omic system biology data and use visual analytical tools. And as we're talking about today, we have phylogenomic and epidemiological analysis for rapid outbreak response. Then as a BV-BRC user, you have a private workspace for analysis, sharing, and publishing. And if you are computationally savvy, we also support APIs, command line interface, and FTP. Then we also want to make sure everyone's using the BV-BRC, so we support outreach and education like this. A quick overview of the services. So we have genomic services, phylogenomic, protein tools, metagenomic, transcriptomics, viral tools, and outbreak tracking. This will all be covered in next week's webinar, but you can see some outputs that you can get from these tools. And we also support surveillance data and the protein structure data. So when we're thinking about the H5N1 outbreak, influenza is a highly contagious respiratory illness, and in 2020, the 2344B lineage emerged and spread through migrating birds in Africa, Asia, and Europe. Then it spread to North and South America. In 2022, other animals like mammals began showing this strain, and as in 2024, we've seen it in cattle and cattle to human transmission. So that's when we began having these outbreak pages to support the understanding and making sure all of the metadata surrounding our H5N1 genomes was accessible and searchable for you. So the BV-BRC is responding to this outbreak by sharing relevant H5N1 news with the community, assisting in the submission of thousands of sequences for 2024, generating phylogenetic trees and continuing to update them as on a biweekly basis or weekly to biweekly. We sent an infectious agent report in our newsletter. Great time to plug our newsletter. If you're not on our mailing list, I recommend joining that so you can stay up to date with what we're doing. We created the outbreak pages, and I added this last bullet to see if there's anything else that you would like to see from the BV-BRC in an outbreak response. Feel free to let us know, and we're happy to try to work with you to make the BV-BRC as useful as it can be. So our outbreak pages follow this format of having an overview with some broad level information, news and publications that are updated in real time. We have an interactive outbreak map that pulls all of the genomic data from the BV-BRC and puts it on a map so you can see what where sequences exist and where data is coming in. Then Christian will talk a little bit more about our phylogenetics, but we have both a segment-based tree and that's curated and generated, and clustered phylogenetic trees that are also segment-based. And the data tab includes links to BV-BRC data, SRA and NCBI virus. And then we have a compiled list of resources to help with analysis and command line tools to bulk download data from the BV-BRC. And we'll look at the outbreak pages on the website, but that's just a quick overview. And I'll let Christian tell you a little bit more about our phylogenetics on the outbreak page. Christian?
Yes. Oh, I was. Yeah. So basically, for the H5N1, we have two set. I mean, we have two sets of different trees, and each tree themselves there's 8 trees. There's one tree for each segment, and so basically the issue was that oh no, I should say something different. So the data is collected. It's relatively complicated. So what we're trying to do is for the 2024 strains, we try to include all the isolates, but since those are they're very similar to each other, so the tree wouldn't be not be very interesting, because basically just a line. So we added older sequences to it, which are mostly manually curated to kind of give you some kind of an overview of certain older sequences. So, and also to root the current outbreak and to give you a sense of proportion, because as I said, the current outbreak, they're very similar to each other, and some of them are even identical. Maybe not on the whole genome, but on the level of individual segments. So and maybe now we could look at one of the unclustered ones. So if you can switch. So that's kind of the live demo.
We can also, we'll let that load and we'll go through the rest of the slides. Oh,
okay. Oh, okay. So, it was just there.
It wanted to. My computer does not love demos and presenting at once, so sorry for that. Well,
yeah, so
we'll put them in the context of the outbreak pages. So I'll switch over to our monkeypox virus outbreak information, and then we'll review both sets of pages in depth. So the monkeypox virus outbreak is starting with mpox being an infectious disease that causes a painful rash, enlarged lymph nodes, fever, headache, muscle ache, back pain, and low energy. mpox is caused by monkeypox virus from January 1st, 2022 to June 30th, 2024. Over 99,000 confirmed mpox cases have been confirmed, and there are 208 deaths globally, over 111 countries. This is due to clade 2b, primarily the global outbreak of clade 2b. Clade 1 has remained a significant concern in Central Africa, and the DRC, being a key region for clade 1, has a continuous rise in cases, and there has been a notable increase in case fatality ratio. So the last thing is there is some concern for clade 1 outside of Central Africa. There's been one case in Thailand and one in Sweden. Just being cautious of outside of the traditional host rate or geographic range. So you can see here one of our outbreak maps that show all of the public genomic data as well as the detection of clade 1 or 2 in different countries. So our outbreak response for MPXV, we shared relevant news with the community, generated the phylogenetic trees that we continue to update, created the outbreak pages, added the monkeypox virus subspecies classification tool. So you can use that tool to analyze any sequence that you have to get that clade designation. And then we classified all of the data in the BV-BRC so you can be able to discern that as you're generating your data sets. And again, if there's anything you want to see from the BV-BRC, we're happy to take that feedback. So our outbreak page is very similar, just no clustered trees, but that will be, as I mentioned, our data is organized in a systematic and structured way across the BV-BRC, so you can expect to see this structure across our outbreak pages. Christian, is there anything you want to say about the phylogenetics here?
Well, here it's in a way this is very different than the one we provided for the H5N1, because those are manually curated sequences, so those are not all sequences. And also, you can see also we have annotated internal nodes with the clade information, and also these trees. We also have a subspecies classification service, and so this tree is being used as a reference tree for the subspecies classification service. Whereas in the case of the H5N1, we have since that, as I said, that tree was mostly focused on the 2024 outbreak, so that tree is incomplete. So this one, so the one we use for subspecies classification it's a different tree. So those are kind of the two main difference. And also, obviously, the monkeypox virus it's not segmented. So here we have these trees for the whole genome. So there's no individual trees for each segments, because there are no segments. So it's just one tree. Whereas for the monkeypox for the H5N1 we have segments. So, but yeah, we should still look at the live trees.
Yes.
So yeah. Now that's the H5. We're going back to the H5N1, so that's the 2024 outbreak. And as you can see here, there's two tabs. There's one, it's labeled phylogenetics, and the other one is clustered. It's labeled cluster phylogenetics. And so yeah, so the one which is just labeled phylogenetics, that's for the full tree. So that's all the sequences for 2024 are in this tree. And you can see it's much too big to be visualized meaningfully, and you can see also the different 2024 sequences. They're pretty much identical, and here you can see, so we have a tree for each segment, so there's a total of 8 trees, and that's, as I said, this around here, those are the older sequences, and of course that's not by far not all the older sequences. It's just the ones I selected to give a little bit of an interesting background to this tree. So you can see some older sequences, but it's not by far not everything. As I said, it's just to give a context. And so maybe that's all I want to say about this one. And then if we switch here to the clustered one, so you see now the current outbreak in 2024 is much smaller, and we still have the same older sequences. And what is the difference here too? Before we had 8 segments, and this one has so we still have the 8 segments, but we have an additional tree for where we artificially concatenated all the segments. So that's then this tree has a much stronger support because the sequence are longer. You can see we can turn on here too, we could see the support values, which are pretty good. And so this tree is made a little bit complicated. So we use, as I said, we use older sequences, and then we use newer sequences where we use a program called CD-hit to cluster it with a cutoff of 0.99, and then we put them together and build a tree. Another difference is for this one here, for the cluster tree, we only use isolates which have all 8 segments. So if you switch from segment to segment, if you look at it in detail, you find all the same isolates on each tree, and then the same thing with the concatenated one. So they're very consistent. So I think that's all I can say here. So we have, as I said, we have two tabs, clustered, unclustered and clustered, and then we have 8 segments. And for the clustered, we also have concatenated sequences. And then now I'm going to show you the monkeypox. Where is it here? So that's the monkeypox. And as I said, this is a little bit different because here we have, this is much more curated, the sequences. And I try to have representative sequences for all the major clades. So we have clade, we see the internal, so we have like clade 1 and clade 2, and then we have, further like clade 2B and 2A and so on. So we can see all those. And but this is not all the sequences, it's just like a representative coverage. And as I said, the main purpose of this tree is actually to be used as a reference tree for subspecies classification. And since I'm already showing it, I can also, we use a program called Archaeopteryx.js for the visualization, which allows you to, for example, you can change the visualization, what you're showing here. Now we show clade, but for example, you could also show years or, other information, the host, which here is not very interesting. We only have 2, human and chimpanzee, and so on. So you can do different things. And as I said, if we can, for example, showing the confidence value, which is pretty high here. And we have like internal nodes. And you can also, that's another useful thing, for example, is if we say this to default, we set this to default, we can, if you search for something, you can enter, like search everything as a D in the name or something like that. And also, so you can search for various things, and then, for example, like, sapiens, and it will highlight those. So you can do that. You can remove it, and so on. So that's the things you can do here. So that's what we use for visualization. And obviously, the focus of this talk is not about Archaeopteryx.js. So I'm not showing all the details, but that's our program. And if people have questions about it, I'm happy to answer more questions about that. I think that's.
You can also download this or any tree in the Archaeopteryx view.
Yes, it's just
the bottom.
the problem. Yeah, and another interesting thing is you can you can kind of compose your own name. So for example, you can put on the host in the name, or host and year, and so on. You can do a thing with the country, the clade. You can put whatever you want as a label for the nodes, and then, as I said, for example, can show confidence values, and yeah, you can download it here as either as a picture or as a in a format like file XML or New Hampshire. So just the screen here is a little bit small, so you cannot, because it's compressed because to the presentation. So, and here that's with this one you control the visualizations. So any questions? Please, maybe you can ask later or put it in the chat box. And I think that's it. So that's what I said. So basically we have trees for both, and monkeypox we have one tree, and for the H5N1 we have two trees. One of them is the full trees and then the cluster trees. Okay.
Wonderful. So I'll go back to sharing, and we'll kind of put those phylogenetic trees in the context of our outbreak pages. So right now you'll see the BV-BRC landing page. We're on a Thursday, so we have scheduled maintenance on the second Thursday of every month. So that is a great reminder that the BV-BRC team is always trying to keep the BV-BRC as useful as it can be and up to date. So it totally is useful and working during this upgrade, but just wanted to let you know that the BV-BRC team is always updating what's going on. So our outbreak pages fit in our outbreak section on the homepage. You see that right here. And they can also be found under tools and services if you've left the homepage. So under this outbreak tracking section, we're going to focus on the Mpox and influenza H5N1 today. So we'll start with the influenza H5N1 outbreak page, and you can see that here we have some background information and news surrounding H5N1. So this news is pulled in real time along with current publications. Can also see the history of all that news. So if this is a great place to quickly get some good information from reputable sources or papers that are surrounding this. As we scroll down through the overview page, we have this outbreak background, our awareness reports. So the BV-BRC is funded through NIAID, and we support NIAID by sending our outbreak weekly report. So if you click here, that will take you to this page, which shows all of the reports that we have generated so far. So the last one we did was on September 24th, and that will show you this report, which gives a good overview of the H5N1 outbreak, what cases have genomic data, what viral sequences we have in the BV-BRC, what the host range for those sequences, and some links to the phylogenetic trees and news and publication that was relevant since the last report was generated. And so these are all downloadable and clickable for you, so feel free to use those as you wish. We also link to some other outbreak awareness reports from the CDC, USDA, and WHO. Then we have the static outbreak map. So this map is tracking any cases of H5N1 in human and cattle. So that will be updated as new cases arise, but is not linked to genomic data. And we have some background in influenza and some epidemiological information. Our outbreak map on this tab is showing the genomic data. So you can see that we have the cases that are in blue are in human or other hosts, and then green will be cases in only non-human hosts. So you can click on any one of these state bubbles and you can slide the map in and out. There are also, it lands you on the US map, but you can also move it over to see the global map. It won't be quite zoomed out like that since I just zoomed it out, but you'll see the world perspective as well as the US. So you can then click on any state and see what the hosts are. And then that will take you to that specific data set. So here we clicked on Wyoming having cattle, and then that will pull you to those H5N1 2024 Wyoming samples. So then you can do something with that data set, and you can get to this through the data searching process that we talked about last week. The slashes are human to other hosts. So in Michigan, we have one human sample and then the other host data. And so you can get looking at all of Michigan or those specific hosts in Michigan by clicking on those, which will then bring you over to the BV-BRC data page, as we just looked at with Wyoming, for example. The one in the middle is all of the US. So you can click here to get all of the country level data. That will always be a diamond. So we saw with Germany, we have the diamond shape for that country level data. So have some links out to data. So you can quickly get to all of the H5N1 genomes in the BV-BRC by clicking here. All of the ones that have GenBank accession numbers. As I said, we have been assembling H5N1 genomes from SRA, so you can get those quickly. All of our bulk data download the FTP site. And then the USD Outbreak Bio Project, raw reads from SRA, and outbreak data on NCBI virus. So these links go out of the BV-BRC, and these ones stay with it. We did a webinar in the May on covering some of the BV-BRC outbreak response. So you can see that in our resources, along with tools you might be interested in using with the BV-BRC, and then other resources that we've collected from other teams out there that are supporting analysis of this outbreak. Our command line tools include some useful commands to support bulk data download. So we have searching for genomes, downloading annotations, downloading genomic data in FASTA format, and submitting an annotation job within the BV-BRC. So these commands are here to make that as seamless for you as possible. And we also have many pages of command line interface documentation, so you can feel free to review that as well. So that covers our H5N1 outbreak pages. If there's anything else you wish to see, as I mentioned, please feel free to let us know. These will continue to be updated as the outbreak progresses. And now I think it's time to look at our Mpox pages. So this page, very similar format, and as I mentioned, you can access it from the homepage or here under Tools and Services. So we'll click on that. This format is pretty similar, the same sort of high quality news, PubMed articles. I think there's too many, so we'll just show more. Having some background. Our maps are a little bit different here. So looking at a global scale, clade 1, clade 2, and both clades. These are also updated as needed. And then this one just overlays that genomic data component as well. We're not currently producing our own outbreak awareness report, but here are some links to some outbreak awareness reports. Information about emerging concerns, epidemiology and transmission, characteristics and diagnostic challenges, timeline, monkeypox virus taxonomy and the genome, from viral zone, and that kind of covers our overview page. We have the same outbreak map, starting you on the global map. So you can see more country level data, and then the colors, oops, wrong scroll, sorry about that. And you can see the key here with clade 1 only, clade 2 only, both country and state level. And this is just for 2020. So our data page includes the outbreak genomes and then the FTP site. Then the same BV-BRC resources and then some other resources and our command line tools with the commands to pull out the monkeypox for our students. And I'm gonna, unless there are any questions, I'll take a second to let you ask if you have them. If not, we'll think about our viral tree service. So BV-BRC has the ability for you to generate your own phylogenetic trees. So you can create custom trees from selected genomes, genes, or proteins. You can take a nucleotide or protein input sequence. There's a fast tree option available, which calculates large minimum evolution trees with profiles instead of using a distance matrix. Oops. We have two maximum likelihood tree building algorithms, FIMO and RAXMO, and those use a substitution model to enhance the accuracy of the phylogenetic topology inferred from nucleotide sequences. And as a user, you're going to define settings to tailor your analysis to your specific data, which we'll talk about as we're submitting a phylogenetic tree job. And once we kind of talk through the service, I have an example that we can talk through. So from the home page, you can see there is the way to access our tools and services. So under the phylogenomics section, we have a viral genome tree or a gene protein tree. Both of these have a similar structure. So if you were going to click on a viral genome tree, there'll be the input, alignment parameters, tree parameters, and these metadata options below. I'm going to reiterate the metadata options many times, just because this is the way that you'll be able to color or designate your tree. So it automatically comes with these features, but you can also add things like any of the metadata features within the BV-BRC. So if you want to be able to color on collection year, you'll have to move that over, for example. So each of our services has some tutorials, quick reference guides, and videos. So these can be selected here. So this will take you to an overview of the quick reference guide to just help you talk through the steps that you need to successfully run an analysis, and the same on the tutorial side, just in a little bit more detail. But if you ever are stuck, definitely don't stay stuck. We're happy to help. We'll also have instructional videos, which will take you to our YouTube. And since we're at our YouTube, it's a great place to see all of the different playlists and videos that we have. So feel free to check these out to get the most out of the BV-BRC. So going back to my presentation, we're going to follow a monkeypox virus example. So we're looking at the OPG057 gene, which is the palmylated EEV membrane protein. And I mentioned that you can get structure data within the BV-BRC. So you can see a predicted structure that took a screenshot from a BV-BRC page of this protein. This is orthologous to the F13 gene in vaccinia. And it's conserved across the subfamily Cordyceps and is critical among orthopoxviruses to produce the wrapped form of virus, which is required for cell-to-cell spread, and it's a target for the ST246 antiviral tecovirimat. So this gene is highly conserved. There are some variants that have been responsible for drug resistance. So just a further view of our structure data, you can see the G277C amino acid change is responsible for drug-resistant phenotype. So this is the without that mutation, but you can see that spot here on the structure view just by clicking that 277G. So no C switch here, but that just showing some of the capabilities of the structure data, which you can access from, for this example, if you go to the monkeypox virus, and I'm going to go very quickly through this since we looked through the data in a lot more detail before. But so you can go to genomes, we'll go to features actually, and we'll get the gene. And then I will select that. And then I'll go to protein structures. And from the structures, you can hit the structure button, and then that will open up the structure to look at, move around, highlight anything as you see fit. Just wanted to show that capability. So that is one amino acid change that's associated with the drug-resistant phenotype. And reviewing Jafar et al., the ST246 is a key antiviral to inhibit viral F13L phosphorylase, one of the essential proteins for orthopoxvirus wrapping. There's this table with a whole bunch of resistance mutations. So you can see the amino acid changes here. If anyone's interested in this paper, you can find it at this link or at this PMID. But as I was putting together a data set, I reviewed some of these positions to see if we saw any changes in the monkeypox virus 2024 sequences. So I took all of the OPG057 sequences from 2024 in the BV-BRC. I ran a multiple sequence alignment and generated a tree. I pulled out some of the positions just so that we could look at those. So for looking at D283, here we can see D283 is completely conserved across this data set, and the same was seen across all of those variants that were associated with resistance across the orthopox viruses. So you can see that there really haven't been any changes in these key amino acids. But when I started to generate a tree for this, I realized that because this was a highly conserved region and highly conserved gene, that any changes were going to be really highlighted on our tree. So the data set isn't very large, and it really highlighted the need for clean data. So because this gene is so highly conserved, we have a few sequences that had a lot of unsure or poor sequence quality. And so those stood out very clearly in my multiple sequence alignment, which brought me to pull those sequences out to remove some of these long random branches to make that a little bit more effective. So we can see that the tree looks a lot more confined when we don't have those long branches. And so when we're thinking about running a phylogenetic tree in the BV-BRC, you can see that you'll run a job, and we'll run through that quickly, but just to round this example out, you can see the clean group tree in real time. Let's take a second to load. And I also just want to highlight that this job is run this morning, so even though our downtime is happening, jobs are still being run. You can see that this gets a little bit more clear. There's still some that need to be looked at further, but very similar sequences across. So if you pull the names away, then quickly see what adding or taking away those facets like Christian was mentioning. And label different colors. Is there anything you want me to show here, Christian? They're all from 2024, so that
you pretty much already showed you can visualize with the box on the right side. You can choose different visualizations which are different from tree to tree. So sometimes you have clade information or host information, and then on the left side you can, as Anna already said, you can determine what's part of the name, and you also have a whole bunch of as you can see, these trees, it's kind of big and there's like overlapping. So what you can do is if you go to the E button, it will expand it so that there's no more overlap. Or then if you go to the F1, that's stands for fitting, so then it fits the whole tree, but you might get overlap. And below that, there are sliders which determine the sizes of various things like internal and external labels, if you have them, the thickness of branches, and so on. And then you have search boxes. You can search in different ways. You can search whole words or part of words. You can even use regular expressions. You can invert your search result. And then below that, you can download your tree again in a variety of formats, as already mentioned. And you also have tools which are kind of in the middle. So you can root the tree, or you can move to subtrees or move back from a subtree. So there's many things you can do. So
And if you click on a specific node,
yes,
you can display the data, go to that subtree.
You can reroute it. You can even delete it if you want to do so. And you see, that's a very weird looking tree, and that's it. I mean, I would not even call it a tree. The fact it's because those sequences they're identical, so that's why you get these weird, like almost like a stair kind of pattern. But it's not. As I said, in this example, the sequences were chosen to be very similar, or I mean, they just happen to be similar, and so or the same. So that's why it looks like that. But most of the cases you will not get something which looks like that. So that's just like some kind of artifact of branch lengths being 0 or near 0.
Yes.
So
and so how we ended up with this very similar tree is that I generated a data set. So I started with the monkeypox virus and I pulled out all of the 2024 sequences. And then from that, I took it and created a feature group. So this feature group was just for that OPG 507. And so you can see these 539 sequences. And then I ran a multiple sequence alignment. So going into our protein tools, starting with unaligned sequences, and can click on that feature group, pick what aligner, stick with that, then just run that alignment, and then that alignment informed my removal of sequences that had, had some sequencing errors. So then that allowed me to make a clean group. And so this feature group has 486 sequences. So then I removed those sequences that had some sequencing errors that I came out of the multiple sequence alignment, and I generated this gene and protein tree. So same view as our viral tree, just for genes and proteins. Can start with a feature group, but we'll start with the FASTA file. So since we aligned it, then that will run the selected feature group. I'll leave the alignment parameters and tree parameters alone. And as I mentioned, it will be important to change the metadata field. So I definitely want to see clades. You have the option of basically any one of the metadata fields that are contained within the BV-BRC. So I'll scroll down so you can see everything. Can add host name. I'm also want to add the clade. And yep, so then those metadata features are added. It won't let you run a job without giving it a name. And then you can submit that tree. And as you run any job in the BV-BRC, you must have a BV-BRC account to run jobs. Totally free to do, but they'll show up here on the bottom bar. So the multiple sequence alignment is running. That will take a few minutes, but we already ran that. This multiple sequence alignment. So that job, I looked before at this alignment file to pull out those to get the clean set, but you can view the multiple sequence alignment here. And it might not load that good as I'm presenting and have all these other tabs open, but that's mostly just the bandwidth of me giving this presentation. But as I said, this gene is very highly conserved. Which you can see just like you're looking at the beginning. Our multiple sequence alignment tool allows you to change the ID type. You can get away from these BV-BRC IDs and change to genome name or clade. So you can see that. And then once the tree is run, just a second. The tree will come with a whole bunch of output files. So there will be an HTML report, text file, Newick tree, Philo XML tree, and an SVG to review in the FASTA. And this view icon will show you the best view of the job output. You can see what parameters you ran the job with. So if you gave it a name that you didn't remember, you can see what trim threshold you used, what your output file is, the tree type, substitution model, and so on. So you can rerun that. You can download any of the data, and then that view feature will take you to the tree. That's just my computer not wanting to participate in this demo. But one other thing that you can do is that you can rerun any job or report any issues, and that if you report an issue, that will give the whole job output. So if it didn't work how you expected, or you just thought there was something missing or anything like that, this will send us a ticket so that we can help answer any questions. So important to have clean data, I mentioned. And just to recap our whole discussion, we talked about the influenza H5N1 and MPXB outbreak pages, in-depth conversation on our phylogenetics, the monkeypox virus example, and just some concluding thoughts now. If you want to dive deeper on anything, we have a Back to Basics webinar series that covers searching for data, annotating viral genomes, building trees, sequence submission, and analyzing protein data. All of the pages have help documents. There are Archaeopteryx help documents, so you can get the most out of that really useful tool. Please follow us on LinkedIn, command line help documents, and we're always here to answer questions and to get any feedback to make the BV-BRC more useful for you. So this is our wonderful team, and thank you for your time today.